Oh, no. Thanks for calling through and all that.
Oh, no. And I'll explain that in a second.
Okay. So, the reason that whole thing happened is apparently, and I didn't know this until recently, your online hours are kept in a separate department from your in-person hours.
Uh-huh.
And so it's not like I don't believe you don't have them.
Mm-hmm.
It's just on my end, the system says there's no hours left, which means
You
I can't do anything.
Yeah.
And so I called like 4 or 5 different people, and I finally got somebody to call me back, and I was like, look, here's the situation. They're like, okay, do it this one time, never do it again.
I mean,
I handle it on Monday. So
Yeah.
it's not that I don't believe you. It's just that
totally
my
understand.
hands are kind of tied. Plus, I hate to say it this way, let's say you call them, and they say, yeah, we're going to migrate those hours over, and they never do.
Uh-huh.
I just wouldn't get paid for my time. And
Yeah,
so that's something I also, no offense, want to avoid.
Of course, I know. But,
So,
I think the fact that you do know that I have hours.
Well, that was the thing is, you said you had hours. The problem is, it's not that I don't believe you. They physically don't show up on my end.
I know.
And so I'm used to students saying, hey, I added 3 hours.
Mm-hmm.
Hey, I added 4
Not
hours.
30.
I'm not used to somebody going, hey, I got 30 unused hours, and I have to turn around and go, I can't. Hold on. Somebody want to tell me what's going on? And so I had to basically get approval.
Mm-hmm.
It's not that I didn't want to do it, I had to get approval. So
Thank you.
No, absolutely.
I was a little anal about this morning because I woke up. I was like, "Are you kidding me?" Because I spoke to the guy.
Did you talk
I
to
didn't talk to Teresa. I talked to the guy, and he said, "Yeah."
Andrew?
Andrew, yeah.
Is that his name? Okay.
Andrew Wallace.
Did he say he was going to move him over? Or?
Yeah. So
Okay.
like,
And the system may have just not even updated yet, and that could be. It could just be everything's crossing through the ether.
as
all
far
at
as
one
you and
time.
as far as you and I stand, how many hours do you want to for your subject, biochem?
Well,
biochem.
that was going to be one thing I did want to talk to you about today. Was of the remaining hours that you're gonna, how much of a chunk you're gonna move over. Now that we've gone through a good bit of stuff, what other things would you like to talk about? Does that make sense?
Yeah, no.
Because if you say I want to talk about these 3 things, and that only takes a half hour, we're just going to do an extra half hour today. But if you say I want to go through all of neuro, all of immunity, and all of this other thing, I'm going to go, okay, we're going to need a couple hours for that. Does that make sense?
I mean, I know. I think you said it's not high yield, but endocrine system.
Okay, we can do that, sure.
Um,
How are you feeling on the nervous system? That comes up a lot.
I mean,
Not as much as the others, but it's still...
I mean, the fact that you say it comes up quite a bit, I would say go over it, even though I think I have a good grasp on it.
Okay.
Just to be safe and sorry.
How do you feel about the circulatory and immunity? Lymphatic, we're not that concerned about. But
Okay.
these two... Because that's the thing. What's problematic is everything you could see. And so as soon as I say genetics and the endocrine system and all these things are high yield, you could get none of that stuff and get everything else because it's random draw.
Yeah.
And so the bio instructor and the chem instructor in me wants to look at you and go, we need to go through all of it. The problem is I know that can't happen. And so I kind of need you to pull me by the ear and lead me in the right direction.
Okay. So let's definitely, today we're hitting...
reproductive and the remaining of the battery stuff that we didn't get to.
That's right.
Last time.
That's right. Okay, we'll hit that today, and then for future tense...
Because what you can do is you can always move a chunk over, and if we don't use it, move it back or use it somewhere else. Or
what's the next high yield that I cover?
So, after what we've gone through,
I know I still somewhat struggle with like polymerases and
right. I was going to say we've gone through that stuff, but I don't know how you still feel about it. If you want to talk about other things,
yeah.
If I had my druthers of we just need to talk about this, we should probably talk micro.
Okay.
We should probably talk nervous, and we should probably talk circulatory and immunity. Just because immunity's much oversimplified for this exam, but the things they do key in on, I at least want to have that
I
conversation.
definitely want to hit micro, yeah.
And so I'd suggest, and given the timing of everything, because I want to talk to you about that too, because I'm completely unavailable the last weekend in March, which is like the 30th, 29th and the 30th, and then like the 1st of April.
Okay.
I'm literally in Houston doing a training, and so I physically won't be here.
Okay.
And so the other thing I wanted to work out with you is you're taking your test April 20th. Okay. So that at least frees up sometime in April should we need to get together. Okay, I didn't know if you were in the early April, like 5th, and then I was going to go, shit, okay, we have one weekend, right? Because I'm open next weekend.
Okay, so let's schedule for next weekend then.
Okay.
I'm meeting with...
So that's the 24th, 25th. Because today's the 17th.
Yes. So I'm meeting with Jameson sometime Saturday, around 11. So 11 o'clock we're meeting.
Okay, do you know if you're going to do an hour or 2 hours with him?
He said roughly an hour.
Okay, so here's my question. Would you rather put like 2 hours on the books for Saturday, 2 hours on the books for Sunday? Because what we've talked about going over is a fairly big chunk of stuff. And you'd rather get the content out of the way earlier than later.
Yeah.
So I'd say we should do a big chunk next weekend.
Okay, so 2 hours.
Would you rather do 2 and 2 Saturday and Sunday, or do like a 4-hour session on Saturday?
Let's not do 4.
Let's do 2 and 2.
Yeah.
Is that but I also didn't want to make you have to buy a hotel room just to see me for 2 hours on Sunday morning. Does that make sense? And so I wanted to throw all those things at you and go, okay, what are you feeling? How does this work?
I'm gonna come in Friday afternoon because I'm gonna see Raheem on Friday.
Okay.
See him from 11 to 12. I mean, we can do 2 hours Saturday, 2 hours Sunday.
Okay.
Yeah.
Sure. That works for me. I just wanted to make sure that pushing the 2 hours to Sunday wasn't then forcing you to get a hotel room for another night and have more expense. And I didn't want to be that guy.
I'll talk to Teresa and make sure those hours get transferred.
Sure.
So Saturday, I'll see you.
Do you want to say noon, and then if Jameson says you're going to run till 1 o'clock, just let me know and we can push noon to 2 to 1 to 3. Or do you just want to set it up as 1 to 3 on Saturday?
Yeah, let's just do 1 to 3.
Okay. So let's do 1 to 3 on Saturday.
And then Sunday, you just want to do 10 or 12?
Hold on, let me pull up my calendar. I'm inputting what the Saturday session is real quick, so hold on.
And do you want to do all bio, or do you want to do bio and chem? Or?
We should probably do bio one day and chem the next, only because an hour sometimes isn't enough to get through all the stuff we need to get through. I can't do 10. I could do 1 on Sunday as well, 1 to 3. Or would like noon to 2 be better? The problem is I'm already booked in the morning on Sunday morning.
So let's do noon to 2.
Noon to 2? Okay.
Yeah. And then do chem, or you want to do, does it matter?
Honestly, it doesn't really matter.
Your call.
6 one way, half dozen the other, right? I'm just going to show up, and whatever we did the day before, I'm going to do the other one that day. So, because that's the thing with me. If you say chem Saturday and bio Sunday, and then I get here and Saturday you say bio today and chem tomorrow, I'm gonna look at you and go, okay.
All right. So bio, we're aiming for micro, nervous, and chapter 9, right?
So yeah, we probably won't get all of that done.
I'm saying it, but I'll have those things covered.
Yes. Yeah. Those are the things we're gonna shoot for your next couple of sessions. So basically, I'm gonna start with neuro. Get as far as we can. If we get to endocrine, I'll hit endocrine and then save circulatory for the next one probably.
Okay.
Because neuro and endocrine, if we can get that done in 2 hours, that'll fill the 2 hours, if that makes sense.
And then for chem, what do you want me to have covered?
Well, that was going to be my next question. Remaining from chem, right? There's the whole thermodynamics chapter that we never touched. Okay. There's the gases chapter that we never touched. How are you feeling? Like
gases, I just want to go over.
Okay.
It doesn't seem like it would take a long time.
No.
For gases. Thermodynamics, just that main equation, how to apply it, when to apply it.
Well, because thermo, you've got delta G, you've got all the different enthalpies and different ways to calculate. Because there's calculation of regular enthalpy, there's Hess's law, there's heat of formation.
Let's do thermo.
Thermo, okay. Yeah. And that's a big one that comes up a lot, and so it's the good thing about that thermo stuff is really getting into differentiating. Okay, when they say this, what the hell do they really mean?
Okay.
right? Because generally I found, and it's something I'm going to want to talk to you about, the way that people view like endothermic, exothermic isn't the real way that endothermic, exothermic works. And so it's good to have that talk about that differentiation.
Yeah.
All right, cool.
Yeah.
And so what'll happen is, have you noticed that when your instructors put their hours in the system, it automatically pulls hours out? I won't be able to put in this session until Monday or Tuesday when they put the hours over. So just to let you know, there's going to be like a 3-day lag time for this one to show up.
I don't know.
It's just an FYI kind of thing, because some students are really
Type A.
about that, and so
I don't understand why though.
Generally, pre-med students are type A students. Because, and I hate to say it this way, it's not a judgmental statement, I'm the same way. I am type A, science geek.
You are type A. Yes.
If I don't get every answer correct, I'm going to be the guy arguing with the professor that you asked this question wrong, right? That's just my mindset. And most pre-meds are that way because the medical profession is very much, you got to know your dosage and the right thing at the right time, and you can't question yourself ever. And so that tends to lead type A's into that kind of, I'm very organized, and if anything's a little off. So, we stopped where? Did we get all the way through galvanic cells?
We did.
Okay.
We're needing electrolytic.
So let's talk electrolytic. Before we do that, if we have that exact same galvanic cell from last time, but let's pretend, and I don't remember which color is which, right? But let's pretend we've run this thing and it's dead. Right, we plate it at the cathode, we have degraded it at the anode. This. Damn, red markers, half of them don't work.
It's not your color.
Let's see. Zinc. So this would be copper, if I remember correctly, and it's the anode. This is the zinc. And that's the cathode. Right, this is copper sulfate and zinc sulfate. So let's just say we run this sucker till it's dead, right? So either I'm out of copper anode or my salt bridge is kaput. Is there any way I could replenish that copper anode?
Replenish,
like, is there a way that I could replenish it without putting in a brand new one? Is there a way that I could build this one back?
You couldn't plate it anyway, could you? No.
I could if I reversed the electron flow and made that the cathode.
Okay.
Right, because the cathode always plates. So if I can reverse electron flow and make them go the other way, right, I can replenish the anode. So we can replenish the anode by reversing electron flow. But there's an inherent problem with that, right? Because if I just send them the other way, what does that make my E naught? Remember, last time my E naught for this battery, we had to reverse one, and it was a total of 1.10 volts, right? But if I reverse the electron flow and therefore have to reverse both reactions, what do I do to the sign of that voltage?
Flip it.
I flip it, right? But that makes E naught cell equal to negative 1.10 volts. And a battery with a negative E naught cell has what kind of delta G?
Negative.
Positive delta G.
Okay.
Right? And positive delta G means it's what kind of battery, spontaneous or not.
A negative delta G?
A positive delta G. Negative E naught cell and positive delta G is non-spontaneous because the electrons don't want to go the other way. So how do I make them go the other way?
Put in a battery.
Hook it up to a power source, right? So this is non-spontaneous and has to be connected to a power source. Well, a non-spontaneous battery with a negative E0 cell or a positive delta G that you have to hook up to a power source, that's an electrolytic cell, right? So, I don't have a big enough board to do it, so I'm going to erase and redraw. But if this is our dead battery, right, we're not going to have the same cathode and anode, but let's just talk about what has to occur. So I've got to hook these things up to a battery, some external power source, right? But if I'm reversing the electron flow, is my zinc now going to get oxidized or reduced? Because remember, where do you start with every battery passage?
You start with...
First thing you write down is what? It was in red up in the corner of the board.
It was like in mine, right? Red. Red.
Anox, red cat.
Anox, red cat.
Right. Oxidation occurs at the anode, reduction occurs at the cathode. So if we're sending the electrons the other way, the zinc that was the cathode and was getting reduced is now going to get oxidized because it'll be losing the electrons. So that's going to be zinc solid to zinc 2 plus 2 electrons. So if it's getting oxidized, oxidation always occurs where?
At the cathode.
At the anode.
Anode, yeah, that's right. Okay.
That makes this my anode, and it makes it my electron source, right? That now means that the copper is the thing that's going to get reduced. And that's what always happens. Where does reduction always happen? Anode or cathode? Yeah, so this is now my cathode. I didn't leave myself enough room. And my electrons sink. Which means my electrons always flow from where to where?
In this situation?
Always, no matter what situation. Anode to cathode or cathode to anode?
Anode to cathode.
Always anode to cathode. Whether it's galvanic, whether it's electrolytic, it's always anode to cathode because the anode's always the side of oxidation is always losing, right? So now the electrons are going this way, which means when we connect the circuit, last time we said the anode always does what when you connect the circuit? Does it plate or does it degrade?
It degrades. It
degrades. So now this zinc electrode is going to degrade because we're taking zinc cations and dumping them back into solution. Well, the reduction is here. What always happens at the cathode? Is it plate or degrade?
Plate.
Always. So we're going to plate this one back up because we're taking the copper anions out of solution, right? Turning them into solid copper. We said which ions always go towards the anode, the cations or the anions? Anions. So the anions come out here. The cations always come out where? At the cathode. So far, has any of the chemistry changed?
No.
None of it. Two things have changed so far. Your sign of E0 and your sign of delta G, right? Because the sign of E0 changes the sign of delta G. The only other thing that changes here is the sign of your electrodes. Remember, we said the way to figure out what the signs of your electrodes are, anthropomorphize your electrons, right? So, are they going where they want to go? No, we have to physically hook them up to a battery and force them to go where they don't want to go, which means we're forcing them to what kind of charge if they don't want to go there? Negative charge. And we're forcing them away from a positive charge. That's an electrolytic cell. Literally, there's no difference between the chemistry for galvanic and the chemistry for electrolytic. The only literal difference is sign of E0 cell, sign of delta G, sign of your electrodes. That's it. Other than that, the chemistry is identical, which is why when we started last time, I started with, okay, every time you see a battery, do it the exact same way. Since the chemistry is identical with those three little exceptions, that means if you do it the same way every time, it doesn't matter if you have electrolytic, it doesn't matter if you have a galvanic cell, they work the same way, so the same process will work for both kinds.
Okay.
Because everyone has it in their mind from college chemistry that there's such a huge difference between the two.
Same, just kind of.
And there's not. Sine of E naught cell, sine of delta G, sine of the electrodes. Other than that, everything else is identical.
Okay.
Which hopefully, if you had trepidation about that, that's now gone.
It is.
Because really they just work the same way, right? Because you all, every time. That is, as long as you can figure out what's getting oxidized and what's getting reduced.
Anode oxidized.
Yeah.
Reduced.
Cathode. As long as you figure out what's getting oxidized, what's getting reduced, you literally now can build everything from that.
Okay.
Does that make sense? Mm-hmm. Questions about that?
In the anode in galvanic cells, what's the charge on it?
The anode on a galvanic cell is negative.
Okay.
Right? Because in a galvanic cell, since it's spontaneous, the electrons are going where they want to go. And so they're going away from negative to a positive. Where an electrolytic, we're forcing them to go where they don't want to go. So they're running away from a positive to a negative, which is why we have to force them to do it.
Okay.
Right? Because honestly, if you have a rechargeable battery, when your battery's in your remote control or in your Xbox controller, it's a galvanic cell. When you stick it back in the cradle to recharge it, you're literally doing this, and it's in an electrolytic cell. All you're doing is rebuilding the anode so that you can discharge it when you put it back in your remote control.
Very cool.
When you go outside and start your car, it's a galvanic cell. When you drive around town, you're recharging your car battery, it's an electrolytic cell. Right? And so quite literally, since you do use these every day and multiple things, they like to bring it up because they can relate it to all kinds of different scenarios.
Very cool.
Questions? You good? All right. One last thing from the electrochemistry stuff. Oh, and one thing I do want to bring up as a potential topic to talk about later. Two things. How do you feel about periodic trends?
Fine.
How do you feel about decay?
Fine.
You're fine with decay. Okay, because realize, as soon as you say, I'm fine, questions are about to flow forth. Okay? So, there are two kinds of decay that have the exact same reason that they go through that decay, and they always produce the same species after the decay is over.
Okay.
What are the two types of decay that have the same problem and always produce the same thing?
Beta.
There are three types of beta.
There's beta emission.
There's beta positive, there's beta negative, and there's positron emission. All three of those are types of beta decay because they all deal with an electron in some way, shape, or form. Right? So there's beta decay, positron emission, right? What's the other one?
There's alpha decay.
Well, alpha is not a beta decay.
Okay, so beta.
Just the three betas, you've got beta, positron emission, and electron capture. Those are all beta decays because beta decays deal with an electron. Because a positron is just a positively charged electron, right? So if you just say beta, that's, although true, it's a little incomplete. What two types of beta always wind up with the same thing because they have the same problem?
Beta positive, beta negative, right?
Positron emission and electron capture.
Okay.
Right?
That's right. Okay.
Because, so, let me see. Oh, okay, this is a good little chart. One of the things that you want to remember is why something will go through the decays that they'll go through. Like something goes through alpha decay because the nucleus is just too damn big.
Okay.
Right? Here, there's just too many, neutrons, protons?
Protons.
Too many neutrons. Right? Here, too many protons.
Okay.
Right? Here, too much energy.
Okay.
So, because it's helpful to know those kinds of things, right? Because if they say, this species has too many protons, you want to instantly go, okay, I got two potential decays that can happen here, right?
Okay.
And the reason I bring it up is charts like that are great, but it summarizes information well, but one of the things you want to do with charts like that is you want to see how things are potentially related to each other.
Okay.
Does that make sense?
Yeah.
Because, and I'm not going to go through the whole chart, I'm just going to do a little bit. Let's say you've got positron emission.
Okay.
Right? Positron emission is because there are too many protons. So since there are too many protons, what do?
You get rid of.
We don't get rid of a proton.
You gain.
I turn a proton into a neutron.
Okay.
And then eject a positron. Does that make sense?
It does.
Because what you do is you take a proton, you turn it into a neutron and a positron.
Okay.
Here's why. Electrons have no mass on the test, right?
Mm-hmm.
So that's essentially just ejecting positive charge.
Okay.
Because with beta decay, with positron emission, and with electron capture, your nucleus is going through what's called a transition. That's a transition, right?
Okay.
But for a transition to be valid, you've got to account for mass on both sides of the arrow and charge.
Okay.
Because if I can't account for both mass and charge, something's jacked up. Something's not right.
Okay.
So that's 1 AMU, that's 1 AMU, that has no mass. Okay, those two account for the mass on both sides.
Mm.
That's positively charged, that's neutral. I've got to still get rid of the positive charge, which is why we have a positron. Does that make sense?
It does.
And so it's those little things like that that the test can refer to that I wanted to talk about if you felt iffy at all about this kind of stuff.
Okay, well, we can briefly.
I mean, it won't take us long. It'll only take us like 20 minutes to go through that.
Yeah.
But I go through all these details with every kind of decay to talk about all the different ways they can come at you with questions about stuff.
Let's do it.
Okay. Do you want to do it in a minute, like today, or do you want to do reproduction today and push this to sometime next week? Doesn't matter to me.
Do you remember? Or do I need to?
You would need to write it down and remind me next Saturday, because otherwise I'm going to look at you and go, what are we talking about? And I'll forget that we had this conversation. Okay. But as soon as you look at me and go, remember decay, I'm going to go, oh yeah, we're going to go through decay, and then I'll just do it. But realize I'm going to teach 7 classes. Yeah. In between now and then, I'm going to forget. The last thing from batteries is Faraday's law of electrolysis. All right. Faraday, kind of a big dude. He's won a Nobel Prize. Mm-hmm. He's the reason you can microwave your food and not kill yourself. Because he invented something called a Faraday cage. You know what a Faraday cage is? Mm-hmm. Have you ever looked at a microwave and the glass door has what looks like chicken wire in it? Mm-hmm. It's because when you close the glass door, that actually creates a metal cage all the way around the microwave. And the microwaves created in the microwave can't get through the Faraday cage and out into your kitchen. Cool. So his Faraday cage keeps the microwaves in the microwave. And if it didn't have a Faraday cage, the microwaves would get out. You'd cook your own face off if you were too close, right? Do you know how a microwave works?
How a microwave works?
Yeah. So how is it I can cook a steak in 4 minutes in a microwave? And you should never cook a steak and ruin it in a microwave. But if I took a steak and just threw it on the concrete outside, it would take all day to cook. Mm-hmm. Why does it take all day outside on the sidewalk, but in a microwave it takes 4 minutes? Mm-hmm. Outside on the sidewalk, you only have UV radiation bombarding that steak from the outside that's exposed to the sun. Mm-hmm. Whereas in a microwave, the microwaves cause water molecules inside the food to vibrate. That's kinetic energy. Kinetic energy releases steam from all food, and you cook your food from the inside out in a microwave, right? And it's all due to this dude. So here's what Faraday's law of electrolysis states, and then I'm going to talk about what that means. Okay. The amount of chemical change is proportional to the amount of electricity that flows through a system. Which means this test can now tell you how much electricity goes through a battery, and then they can ask you, how much copper do I degrade? How much zinc do I plate? Right? Because he had a constant. Remember, we talked about his constant, Faraday's constant, right? 96,500. And it's what we're going to do is take one of your physics formulas and rearrange it. Right? Here we have Q equals IT. Yeah. I think in the physics book, it's current equals charge over time, I think. Right? But this is, right, your charge in coulombs. I is your current in amps, right? T is your time in seconds. And what this equation allows you to do is if they say, we apply this much current for this much time, how much copper or zinc do you plate or degrade? Mm-hmm. This is how you get there.
Okay.
And it's all due, right, to Faraday's constant, which we said was 96,500 coulombs per mole. And that's the charge of 1 mole of electrons. So, right, what they could do is they could say, okay, you apply 2 amps of current for 16 minutes, right? Let's say it's that same battery we've been using, right? How much copper is plated? That's all they give you. And so you've got to immediately jump to this, where you have Q equals IT. So you've got to figure out your total charge. So your current is 2, your time is 16, right? But standard unit seconds, so we've got to multiply this by 60.
So 960.
1920 is what that is.
Or 1920.
Right? 2 is 32
Well,
multiplied by 60. Oh, yeah, that. And then double it.
Okay.
Right? That's 1920 coulombs. But Faraday's constant tells us the number of moles of electrons because we know what the charge of 1 mole is. So if I take this and divide by 96,500 coulombs per mole, I know how many moles of electrons flowed through that circuit.
So why did you divide by Faraday's constant?
Because that's the total charge.
And you know that one...
And I know that this is the charge of 1 mole, so by dividing them, that tells me how many moles of electrons flowed through the circuit.
Okay.
And since I know for an oxidation reduction reaction what the mole-to-mole ratio between them is, I can then convert.
Okay.
Right? So what you want to do here, that math, right? So we're going to use 2,000 over 100,000. That just makes my math easier because I can just take 3 zeros off both and dump them. And that becomes 2 over 100, which is 0.02 moles of electrons. That's how many moles of electrons physically flowed through the circuit, right? So if I had 2 moles of electrons flow through the circuit, the big question becomes, how many moles of copper does that produce?
So you give 0.02 moles flow through the circuit. How many?
Would I produce 0.02 moles of copper, or would I produce something else? I produce exactly half.
Half of that. Why?
Because we know for every 2 moles of electrons per this oxidation reduction reaction, I can only form 1 mole of copper. So for every 2 moles of electrons I use, I can only make half as many moles of copper.
Okay.
So if I use 0.02 moles here, I can only form 0.01 moles copper, right?
Okay.
So this is now 0.01 moles copper. So you take the moles, multiply it by its molecular weight, 63.5, and you get 0.63 grams. Does that make sense?
It does.
And so really, Faraday's law of electrolysis, when they say the amount of chemical change is proportional to the amount of electricity that flows through the system, really that's just a really fancy way of saying, hey, here's a conversion to get from here to here.
Okay.
Does that make sense? Do you have your ICC on you? If you don't, it's okay. I just want to point something out. Because basically I just did the ICC question,
Huh.
but I want to point something out to you here. Because this is the shit that the test will do. Where is it? There it is. See, that's the exact same question that we did.
Mm.
C is the correct answer.
Mm.
Notice what D is.
1.26.
D is exactly double what we got. D is what you get if you don't take the 2-to-1 mole-to-mole ratio into account. D is what you get if you use 0.02 to multiply by the molecular weight.
Mm.
That's the kind of shit the test does, right? So that's the kind of stuff you've got to be on the lookout for. So when I say they tend to know the types of mistakes people make, they definitely do. And so if you forget to take the mole-to-mole ratio into account, that'll be an answer choice, right? And so it's those little things, and there's a lot of them. That's part of the big beast of this test. There's so many little things that you got to remember you look out for along the way. Does that make sense?
It does.
And so that's how you're going to use this.
Okay.
Good?
Yes.
All right, cool. Now we're moving into reproductive stuff, because that's really the end of electrochemistry. And so where we need to start is really in the beginning. Right? Reproductive development. This isn't how does a baby develop. This is why do girls become girls and boys become boys in the womb, right? So early embryos, what we mean by early is less than 7 weeks, have undifferentiated gonads and contain both Wolffian ducts, and Wolffian ducts develop into male genitalia, and Mullerian ducts. And Mullerian ducts develop into female genitalia. And the big thing here is knowing the actual pathways of how to get from beginning to end for both boys and girls. Now, the female pathway, super straightforward, right? Anybody with 2 X's, it goes the same way every time. The 2X genetic combination, right, this acts upon the Mullerian ducts. And the Mullerian ducts develop into internal genitalia. All right, we've got to distinguish between the internal and the external, right? Internal, ovaries, fallopian tubes, uterus, things like that. The external genitalia develops all on its own. Labia, things like that, right? Fairly straightforward pathway. There's not a lot to it. When it comes to the boys, very, very different, much, much more complex. And really the complexity comes from that Y chromosome, right? That Y chromosome leads to the development of the testes. That comes strictly from the Y. And what the testes does, right? The testes secrete testosterone. And then also make sure you leave a big gap here, because we're going to fill in a second half of this. We're just not there. Now, the testosterone acts in 2 ways. It can act locally and systemically. Systemically just means body-wide, right? Locally, it acts upon the Wolffian ducts, and the Wolffian ducts develop into the internal genitalia. Systemically, that testosterone is converted into a completely different thing. It's converted into dihydrotestosterone. Or you could see it as DHT, either way. Now, dihydrotestosterone is what leads to the development of the external genitalia. But the testes also secrete something called Mullerian inhibiting factor. Or you could just see it as MIF, right? And what this Mullerian inhibiting factor does is it leads to Mullerian duct regression. All in all, a much more complicated pathway, right? Now, the big takeaway, though, is what is the difference between them? Really, there's only one thing. What's the big difference between these things? We have the presence of testosterone and the absence of it. And really, that's the big difference between developing into a boy or developing into a girl. Because if someone is male and has testes and doesn't produce enough testosterone, this way kicks in, right?
When do you just say, oh, okay.
This is the default biological pathway. And I'm sure that you've heard someone say somewhere in your college career, well, we're all female in the womb.
Yeah.
No. This is what they mean, that with a lack of testosterone, you develop down the female pathway. But it doesn't mean everyone starts off with a uterus, a vagina, fallopian tubes, and ovaries that then turn into something else. No. What they mean with everyone's female in the womb is without enough testosterone, you develop along the female pathway, right? So like in movies, and my students have actually asked me this in class, like, why do guys have nipples? We don't breastfeed. You know why?
Why?
Because we all start off in the same place, and everybody's got nipples in case you develop along the female pathway. They just don't go away if you develop along the male pathway, right? So, like, and I don't know if it was Meet the Fockers or Meet the Parents, when Robert De Niro says, I have nipples, Greg, could you milk me? Given enough hormones, yes. Yes, I could milk you, Robert De Niro, as long as I fed you with enough hormones for about 6 months beforehand. Right? So everything goes here without enough testosterone.
So if you have the XY chromosome, you are in the path to become male.
But not guaranteed. But without, you're not guaranteed. Yeah. Now, and it's not going to be a fully female pathway, because think about it, things are going to get screwed up. The testes produce the testosterone, but if the testes don't produce enough, you're going to develop along this pathway. Kind of, right? We say that, but you're going to have some female structures. You're still going to have the testes, because that's what should have produced the testosterone and didn't, and so things just get all kinds of screwed up.
So you could potentially, let's say you make it down the pathway, and then testosterone's at a halt. Could you have both sexual organs? You
potentially could, but they if they're going to do something like that, they really are going to get into detail with developmental pathways and at what point do you stop testosterone and this happens. Okay. And so what they could do, we run this experiment and we found out that testosterone, if it stops after day 6, does this. Okay. If the testosterone stops after day 12, this happens. Day 15, this. They'll have to give you a bunch of information if they start asking you specific questions like that. Does that make sense? Now, do you want to go through the reproductive organs and what they do? It's just a big chart. The information's in the book. It's not in a chart in the book, but the information's there. So I didn't know if you wanted me to just redo it or if you wanted to read it at home. I don't know how you feel about all this reproductive stuff.
I mean, it seems like you'd simplify it, so let's do it.
Okay, no, that's fine. Plus, even if we didn't talk about it, there's something that I wanted to talk about that I was going to make you talk to me about anyway. So we'll just go over it now. So we've got to start comparing reproductive organs. So we've got to go, we're going to do female stuff, and we're going to talk about the primary reproductive organs first. And essentially what I'm going to do is break it down in a chart of female on one side, male on the other. Okay. Just makes things easier. So primary organs for females are ovaries. There are two cell types in the ovaries we're concerned with. There are granulosa cells, and there are fecal, with a TH, cells. Fecal cells handle oocyte maturation. Fecal cells are all about hormone production. And when it comes to females, the ovaries are the primary organ of reproduction. That's it. After that, they become secondary. But just for comparative purposes, we're going to talk primary, girls and boys, secondary, girls and boys. So primary organs for guys are the testes. Right? They are the analogous structure to the ovaries. Here's an interesting question. Internal organ of reproduction, external organ of reproduction. Internal. Why? Because they're outside the body. So shouldn't they be external? I don't know. You were right with internal. Mm-hmm. Everyone assumes testicles or testes are external organs of reproduction because they're outside the body. Mm-hmm. But remember, where do the ovaries start when you're developing in the womb? Internal. Yeah, they're up here. They are where the ovaries are, right? Given the right hormone cocktail, you can turn ovaries into testes and testes into ovaries, right? They can go either way. And since they start in the abdomen and then have to drop, they're still technically internal organs, right? These are contained in the scrotum. Why are they contained in the scrotum? Why are they outside the body? There's a specific reason.
To maintain temperature.
Temperature, yeah, it's temperature regulation. Because sperm maturation occurs a few degrees below body temperature. Okay. And so if the testes were up in the body, it'd be too hot for sperm maturation, and we wouldn't be here as a species. Quite literally, 3 degrees can make all the difference in the world. Yeah. For lack of a better way to say it, the testes are essentially, quite literally, wound up balls of tubes. Right? Take a tube and just wind it all over itself. That's what a testy is, right? And that tube system specifically are called seminiferous tubules.
This is still primary, right?
Yes. So testes are the organ. The seminiferous tubules are the tube system that makes up the testes, right? And so if this is the tube system that makes up the testes, there are two cell types here that we're concerned about, right? There are Sertoli cells, and Sertoli cells specifically handle spermatogenesis, right? Making of sperm. And these are stimulated specifically by FSH. Easiest way to remember it. S for Sertoli, S for spermatogenesis, S for FSH. Keep all your S's together. The other cell type in the seminiferous tubules are Leydig cells. Leydig cells specifically produce testosterone. And Leydig cells are stimulated by LH. Again, keep all your L's together, Leydig and LH. It's not quite as good because it's not less testosterone, but still works.
Okay.
So that's just the testes, right? I'll start numbering the male primary organs because we've got several. After the testes, what do you think's next? Vas deferens, epididymis, ejaculatory duct, urethra. It's the epididymis. Right? The epididymis, this is the site of sperm maturation. After the epididymis, we have the vas deferens.
What's the mnemonic for it?
I don't have a mnemonic for it. If somebody else does, I just don't know it.
Okay.
I just have everything memorized. Now, you could also just see this called the ductus deferens, same thing.
Okay.
But a vasectomy, here, vasectomy. And they don't remove, and that's the strange thing, they don't remove the vas deferens. They just snip it, they cut out a portion about that big and just tie off both tubes at each end, which is how you can have a vasectomy reversal. They just untie the tubes and connect them back together. Yeah, they don't pull the whole thing out, they just clip it. And the vas deferens, this is the site of sperm storage until ejaculation. Now, this is going to be an odd question. Go with me for a second. Assume a normal, healthy male.
Okay.
How many testes are there? Normal, healthy dude. Two. Is there one epididymis or two epididymi?
I'm gonna go two.
Two, because each testicle has an epididymis loosely attached to the outside. One vas deferens or two?
Two.
I say it's one. Who's right?
Here,
we both are. Here's what I mean. Two of these. This begins as two tubes that join together to form one tube.
Okay.
So don't let them get you with numbers on a thing like that.
How would they get me with numbers?
They could, it could be in the answer choice where they refer to multiple vas deferens or one.
Right.
Depends on how they worded it, right?
Okay.
If they use epididymis singularly referring to an entire male, that's not right because there are two of them.
Okay.
Right? So two of these starts as two tubes, ends as one tube.
Okay.
So don't let them try to fool you with plurality of things.
Okay.
Right? Because honestly, whether they include an S on the end of a word to make it plural or not can quite literally be the only thing that makes an answer choice right or wrong.
Okay.
And so that's why I say don't let them get you with numbers or plurality.
Okay.
Right? Similarly, you have two ureters, you have one urethra. What they'll do is they'll take an answer choice and take the S on ureters and move it down to the urethra so that ureter is singular, urethros is plural.
Wow.
That's completely incorrect. And that'll be the only thing that makes that answer choice incorrect, is that S in the wrong place, right? So this is why I say they suck, and watch out for plurality on things. After the vas deferens, we have the urethra. Right? It's a shared urogenital organ. Really all we care about for reproductive stuff is that this is surrounded by spongy erectile tissue. That's really all we care about. You don't even have to know the names of the erectile tissue. Just know it's surrounded by spongy erectile tissue. So, primary organs for girls, just the ovaries. Primary organs for guys, it's the entire tubular passage from inside the body to outside the body, right? That's how you want to think about it. Now, when it comes to the secondary organs of reproduction, here's where we've got to pick up everything else. So after the ovaries, we have the fallopian tubes. Right? And the fallopian tubes are specifically the site of fertilization, right? Specifically where it happens. The fallopian tubes have finger-like projections called fimbriae. Fimbriae are essentially, think of them as giant cilia.
Okay.
It's literally what they are. So here's the question. You've got to know why the fallopian tubes have fimbriae, right? Mm-hmm. Pretend that my fist is the opening of the fallopian tube. They have giant cilia on the end of it. Mm-hmm. Why do they have these giant cilia on the end?
To keep it held together. I don't know.
Because the egg and the fallopian tubes physically aren't attached to each other. There's literally a space. So if my fist is an ovary, the fallopian tube is like this. They don't touch, right?
Fimbriae.
And so the fimbriae beat and create a current to help draw the egg into the fallopian tube. And that's what they're for, right? These create a current to draw the egg in. That doesn't always work, though. Do you know what they call it if you've got a fertilized egg outside the female reproductive tract, what that's called? It's called an ectopic pregnancy. Okay. Very bad, right? Ectopic pregnancies always have to be terminated. Because if it's outside the female reproductive tract, that means the egg is going to try to implant on the outside of the uterus. Super bad. On the front of the abdominal wall, that's even worse. Sometimes the egg will try to implant on the bladder. That'll kill the mother in about 3 weeks, right? And so no ectopic pregnancy is viable once it gets outside the female reproductive tract. Okay. So that's why the fimbriae are there. Okay. Now, after the fallopian tubes, right, what's your next organ? Do you know? Next, you have the uterus. This is essentially one big muscle. It's essentially all it is, one big tube of muscle. Okay. But there are two parts you have to know. There's the myometrium, and the endometrium. The myometrium itself, anytime you see the prefix myo or sarco, means muscle. Okay. Right? This is just a thick muscle layer. Right? And it's smooth muscle. Right? During pregnancy, the contractions, that's the myometrium just squeezing as much as it can. Okay. The endometrium, this is what the fertilized egg implants in. This is what the placenta grows into. All of the endometrium is essentially for fetal nourishment. And lastly, the last organ we're concerned with for the exam is the vagina. There's only one characteristic here that we care about. This is an acidic environment. The vagina's pH is specifically kept low as a protective thing, right? That acidic environment hopefully will shut down any kind of bacterial infection or fungal infection that could potentially get in there and root, right? Take hold. That's what the acidic environment is for, to protect against infections. Doesn't always work. That's why it's there. Do you mind if I erase the dude stuff? Because we got to put some more stuff over there. Now I'm just out of room because it's a small board. So the secondary organs for guys, there are 3 of them. So the secondary organs here, we have the, it's not the right one. We have the seminal vesicles. We have the prostate. And we have the bulbo-urethral glands. All of these do the same thing, quite literally. all of them produce seminal fluid. All right. Interesting question, but it's a question I have to ask. What's the difference between semen, sperm, and seminal fluid?
Semen, sperm, seminal fluid. The state at which?
Semen is the combination, sperm and seminal fluid. So sperm are the physical swimmers.
Okay.
This is the fluid that they're in. Put all of it together, semen.
Okay.
Right? Semen doesn't mean sperm. Semen is sperm and seminal fluid. Right? Your sperm is just the swimmers. Semen has the fluid in there as well.
Okay.
Right? Small difference in definition, but it can make a difference, right?
Okay.
So each of these produce a different amount of seminal fluid, and the seminal fluid that each of these produces has a different quality to it, and that quality matters. Here, it's about 60% total volume of semen. Here, it's about 35% total volume of semen, and here, it's about 3%. 60, 35, and 3 is only 98%. What's the other 2%? Sperm. Right? These are semen volumes.
Okay.
And so the other 2% of semen is the actual swimmers, the sperm.
Okay.
Right? So the seminal fluid from the seminal vesicles, right? This contains fructose. Why does semen need to have fructose in it?
I don't know.
Energy for sperm. How big are sperm? Really big, really small?
Really small.
Really small. Well, if they don't fertilize the egg till the fallopian tube, comparative to their size, how far is the trip up the vagina, up the uterus, all the way up the fallopian tube to find the egg?
Around the world.
That is a very long way, right? They need energy. This is a crass way to put it, but quite literally, road snacks, right there,
Okay.
right? That is what they use to produce energy for the very, very long trip they've got to take.
Okay.
The prostate, the seminal fluid from this, right? This provides alkalinity. Why do we need semen to be alkaline?
Because it's an acidic environment.
Because if seminal fluid didn't have alkalinity, the vagina would kill all of the sperm, and once again, we wouldn't be here as a species. So the bulbourethral glands, here this is a little different. This provides alkalinity and lubrication. And this is one of the reasons why I wanted to potentially talk about this, is for this differentiation alone. It's really specific and important. These two alkalinities are for completely different purposes.
Okay.
Right? So the seminal fluid from the bulbourethral glands, that's the first bit of any ejaculate, any semen that hits the urethra. The first part of it is always from the bulbourethral glands.
Okay.
Well, your urethra is a shared urogenital organ. No matter how much people think it's gross or don't want to admit it, after you urinate, there's always minute quantities of urine in the urethra. Well, urine is sterile unless something's very wrong, but urine is slightly acidic. So you need this alkalinity to both combat the acidity of any leftover urine in the urethra and lubricate the urethra for the expulsion of sperm. Right? So this alkalinity is specifically for the vagina. This alkalinity is specifically for the urethra, right? That's why it's such a little bit.
Okay.
And so that's why the two alkalinities are completely different. One's for the vagina, one's for any minute leftover urine in the urethra.
Okay.
Does that make sense?
So it aids.
Well, most of the alkalinity here is neutralized going through the urethra.
Okay.
Right? That's why this is 3% and that's 35%.
Okay.
Mainly the prostate alkalinity combats the acidic environment of the vagina, because the alkalinity from the bulbourethral glands is used up generally by the time you make it all the way through
I know this is a personal question. Probably not, because you know all the sciences. But can you get a girl pregnant from pre-cum?
Yes.
Okay.
It's possible.
Okay, because there's leftover.
Well, it's because, although this is the first bit of fluid to hit the urethra, there are always some sperm in there too.
Okay.
Right? Because everything is mixed together, it's just that this is the last bit to join everything, so it's the first bit that hits the urethra.
What's the life rate like once it, I guess, exits?
Now, I have no idea what a half-life is. I can tell you that sperm are motile or active for about 2 or 3 days.
Okay.
But realize changes associated with ovulation can actually make the female reproductive tract more conducive to sperm survival and can turn survival from 2 to 3 days into 5 or 6 days, depending upon where a girl is in her menstrual cycle.
Okay.
So the answer is yes, but it's harder, but it can happen, right? Because honestly, in the moment, how are you going to tell what's the difference between pre-ejaculate and ejaculate? You're not, because you've got milliseconds between the different. Essentially, during the actual sexual act, they're the same damn thing, essentially, right? Questions about this stuff? All right. This is where we have to start talking about spermatogenesis, oogenesis. It's meiosis, but yet not meiosis at the same time. And I'll explain what I mean. So, with spermatogenesis, we're going to start with an initial cell. This cell is a spermatogonium. This cell can mitotically clone itself, can go through mitosis. It can enter meiosis. Here's the deal, though. Since it could do either, if it enters meiosis, we're going to give the cell a different name. It hasn't divided yet. Nothing is technically changed. But once it's going to enter meiosis, it physically can't go through mitosis anymore, so we have to give it a different name.
Okay.
Right? This is a primary spermatocyte. After the first meiotic division, you have 2 secondary spermatocytes. After the second meiotic division, you have 4 cells called spermatids. Then those spermatids are going to physically mature into that sperm shape you recognize as a sperm. Right? And these are spermatozoa.
Okay.
So all in all, this is essentially meiosis. 1 cell to 2, 2 cells to 4, and we're done. Right? But just like when we did genetics and had to keep track of N and X, number of chromosomes, number of chromatids per, you've got to keep track of that here now too.
Okay.
So here's to test the memory. What's the N and the X on this cell?
2 and 2X.
It's 2 and 2X, perfect. What are these? Are they 1 and 2X or are they 2 and 1X?
1 and 2X.
Perfect. They're 1 and 2X. And these here and here are 1 and 1X. Fairly straightforward. This is essentially meiosis. Couple of things we've got to talk about.
Can I write that down?
What?
Or are you going to add more to it?
I'm not going to add more to this. I'm just going to write interesting points that you've got to know over here. Actually, while you copy that down, I'm going to the restroom.
Okay.
Good time. Sorry about that. First things first. When does this occur in males? When do we actually produce sperm?
When do we?
Yeah, so like when in your lifetime? From when to when?
Right around 14, 15.
So what do we call that? Puberty.
Puberty. Okay.
Until when do you stop producing sperm?
I don't know.
You don't. You die.
Okay.
Right? So here, right, this occurs from puberty until death. In fact, men can actually make sperm for an hour or two after death. Because think about it. When your heart stops, does every cell in your body immediately stop at the same time? No, as long as they have enough nutrients and resources to keep going, they will keep going until they run out of nutrients and resources. So, yeah, you can actually produce sperm for about an hour-ish after death. Now, men produce millions of sperm a day, which is why we have to be able to mitotically clone that cell. Because if we were producing millions of sperm a day from puberty until death, which is potentially 70, 80 years, yeah, if we couldn't mitotically clone these initial cells, we'd run out before we got to the age of 20.
Okay.
Right? Now, the other thing is, sperm have one job. Literally one job in the world. Their whole job is to carry DNA. It's their only job. Get the DNA to the egg. That's it. Questions about that?
So?
For spermatogenesis, yes. This is straightforward pictogram of meiosis, right? When it comes to oogenesis, it's meiosis, but it's way different than this. So I needed to put this up there to then say, this is the straightforward one. It's in your book. Make sure you read it. Make sure you check it out. And then we're going to do oogenesis. And the pictogram we're about to put up here isn't in your book. They describe everything in words, but they don't give you the picture, which I've been complaining about for 12 years because they give you the picture of the straightforward one and don't give you the picture of the one that's all messed up. So, quite literally, when we go through this, if you've got questions, now's the time, because what we're about to do, although it's technically in your book, it's not like this in your book. Does that make sense?
And pronounce again, oogenesis?
Oogenesis, yeah.
Not oogenesis?
It's not oogenesis, right? The good thing is, the names of the cells carry over. They're pretty much the same.
Okay.
So we're going to start with an initial cell, but instead of a spermatogonium, it's an oogonium. Right? This can go through mitosis, or it can enter meiosis. Right? Just like before it enters meiosis, it can't then enter mitosis, so we have to give it a different name. This is a primary oocyte. In fact, this primary oocyte, we're specifically going to say is frozen in prophase 1. Because this starts in the womb, right? This begins, of course, begins as an E, not an I. Hi, I can spell. I'm an adult. This begins in the womb and pauses. It doesn't stop. It pauses at about 3 months after birth. And 3 months after birth, at birth, same thing, right? Every single oocyte is frozen in prophase 1. when it pauses right here. That means that females are quite literally born with every oocyte or every egg they will ever have. Once this system pauses at birth, these steps never occur again.
So when they're born.
So at birth, everything is here, and this will never happen again.
Okay.
Right? So quite literally, a female is born with all of the oocytes or eggs she will ever have.
So why is mitosis an option? Or why is it a possibility?
Because you still, even in the womb, need to clone a bunch of these to produce the amount of oocytes women need available for all of their life.
Okay.
So it pauses here at about 3 months after birth, essentially.
Mm-hmm.
The whole system starts again at puberty. So this starts again at puberty and occurs until menopause. And I'm going to ask lots of questions here because, and here's why I do it. Guys don't deal with this. Guys don't deal with the menstrual cycle. A lot of this stuff isn't taught very well in bio classes. So guys tend to know a lot less than the girls do. And I'm trying to make sure I get your content up to where it needs to be.
Okay.
So first question, what does menopause mean? Does that mean a woman is out of eggs?
No.
No. Perfect answer. That's a really common misconception. People think menopause means out of eggs. No. Right? This just means no menstrual cycle for 12 consecutive months. That's it. So that means a woman can be menopausal for 15 years. If she has a random spotting event, that's a menstrual event. That means she's no longer menopausal for another year. Does that make sense? And so it's those little variations in definitions that can make all the difference in the world.
Give me 1 second.
No, please. If I need to slow down, tell me that too.
So I'm going to recap, tell you what I understand.
Yeah, go ahead.
So your oogonium and your primary oocyte, that is made prior to birth, and once they're born, that's all they...
Yeah, everything's frozen right there at birth.
And it gets unfrozen once...
Yes, we start again at puberty.
When you say you start again, what does that mean?
So the system paused at birth, so the system picks back up at puberty, right?
Okay.
So fertilization to birth is the first part of it. Then it pauses for 15 years, and at puberty, the system picks back up, and we've got to go through the rest of oogenesis.
Oh, okay.
We haven't even completed oogenesis yet.
Okay.
All we've talked about so far is quite literally to this point, and that's it. So we go through the first meiotic division, but here it's an uneven division. One cell gets half the membrane, half the genetic material, most of the cytoplasm, and all of the organelles. One cell gets half the genetic material, small portion of the membrane, small portion of the cytoplasm, and that's it. Okay. Uneven. The thing that gets half the genetic material and nothing else, this is called the first polar body. The cell that gets half the genetic material and essentially everything else, that's a secondary oocyte. Does that make sense?
Okay.
So the second meiotic division, meiosis 2, is quite literally the same. It's the same uneven division where one cell gets almost everything, one cell gets almost nothing. The cell that gets almost nothing is the second polar body. The cell that gets almost everything is an ootid, which then matures into an ovum. And this is what I mean by, yes, this is meiosis, but this is not a standard meiotic division.
Why is there...
We're going to talk about all the funkiness and intricacies going on. Couple of big things. First big, huge thing. This second meiotic division, right there, this only occurs upon certain conditions. Meiosis 2, this only occurs upon fertilization. Right? So if there's no fertilization, there's no second meiotic division.
Okay.
Which means, and this question actually pops up fairly frequently, which of these cells is lost during menstruation?
Secondary reason.
Yeah, this is lost during menstruation because if menstruation is occurring, there was no fertilization, meaning there was no second meiotic division. Does that make sense? Mm-hmm. That question comes up a lot because everybody wants to jump straight to ootid, but that never happened, right? So, another little fact over here, the secondary oocyte is lost during menstruation.
Define menstruation one more time.
Bleeding.
Bleeding?
Yeah, the 7 days of losing the menstrual lining, that's menstruation.
And they shed their endometrium. Is
That's menstruation. Shedding of the endometrial lining is menstruation.
Okay.
Be careful. The test likes to use a word that everyone hates. They tend to use the word slough. The endometrium is sloughed during menstruation. SLOUGHED.
I already hate it.
I tend to use the word shed because anytime I say the word sloughed, my students immediately shiver and tune me out for the next 30 seconds, right? So, first things first, let's talk about these two polar bodies, right? They have one job. Polar bodies get rid of DNA. Their whole job is to just get rid of DNA. We're going to talk about why in a minute. They're either shed, possibly, or they're just absorbed back into the body, right? We don't do anything with polar bodies.
Okay.
But don't let them get you with numbers, and here's what I mean, right? First polar body does not go with primary oocyte, right? The primary oocyte becomes the secondary oocyte and the first polar body.
Okay.
Does that make sense? The second polar body comes from the secondary oocyte.
Okay.
But what they do is like to ask things like, what are the products of the first meiotic division for oogenesis? Mm-hmm. And they'll have answer choices that say, first polar body and primary oocyte, second polar body, secondary oocyte. Because people naturally want to put 2 and 2 together and 1 and 1 together, even though they don't really go together. Does that make sense?
That makes sense. Mm-hmm.
The other thing I want you to watch numbers on, each polar body gets rid of half the genetic material from the cell before it, right? Mm-hmm. Which means each polar body gets rid of a different amount of total genetic material.
Okay.
Each of these gets rid of half the genetic material from the cell that came before it. Mm-hmm. That means this gets rid of 50% total DNA. That gets rid of 25 because it's 50 of the 50. But once again, that's one of those things under the anxiety of the test that can bite you. So just don't let them get you with numbers. Okay, now, and I know this seems obvious, but here with this process, we wind up with 1 gamete, where spermatogenesis we wound up with 4.
4,
right? Yeah. This produces 1 gamete, not 4. And that's why polar bodies get rid of DNA, right? Because, think about it, how hard is pregnancy on a woman's body of just 1 fertilized egg?
Hard is it?
Is it tough on her body or not so tough?
Not so tough.
Oh my God, pregnancy's awful on a woman's body. It's very, very tough on a woman's body. It stresses all her systems. Think about it. Even just how do you make room? Although, you get pregnancy belly, it shoves all their organs up and all their organs down to make room for the baby. Like, back hurts. I mean, pregnancy is very tough on a woman's body. Imagine if she ejected 4 healthy ova that could get fertilized every month. Yeah, no, we're not having a litter of puppies in a cardboard box underneath the basement stairs. So, we're striving to only get one viable gamete because that lessens how hard it is on the woman's body. And if we're striving to only get one, I got to have things get rid of the genetic material at each division.
Okay.
So these get rid of DNA so that we can only wind up with one at a time.
So like twins.
Depends on what kind they are. There's fraternal twins, there's maternal twins. Sometimes twins are 2 different eggs got ejected and they were fertilized by 2 different sperm.
Okay.
Sometimes twins are 1 egg got ejected and then in its divisions of becoming a baby, something funky happened and it split into 2 separate beings from the original 1 sperm, 1 egg.
Okay.
Then there's things like chimerism. Do you know what that is? If someone is chimeric, generally it means they were twins in the womb and one of the twins absorbed the other one. People with chimerism have 2 complete sets of genetic material that are different.
Say it one more time.
Someone with chimerism or a chimeric person, if I'm chimeric, I have 2 complete and different sets of genetic material in my body because I absorbed my other person in the womb. And there was they used it on CSI, but it came from an original case where there was a murder case and they thought it was the husband or the boyfriend. And they took a DNA sample and DNA sample ruled him out. He was not the killer. Then they went back and took a different DNA sample like 2 months later, and the DNA matched the killer's DNA because the dude was chimeric and had 2 completely.
So only one of them showed up.
Because not every cell has both sets of genetic material.
Holy crap.
Some cells have 1 set, some cells have another. So you can literally draw a chimeric person's blood on 2 different weeks and get 2 completely different genetic sequences.
Blood types and 2 different.
They won't really have 2 different blood. It'll be like the blood type of the twin is this, and this of the other one is this, and this of the other one is that, and so potentially you're going to have the same blood type, but you can have different genetic elements.
Very cool.
Like once you start getting into genetics and genetic disorders, like there are so many little bitty funky little genetic disorders that can come from multiple places. But that's why we have to get rid of DNA, so that we can only wind up with 1, right? And this is what I mean with, yes, this is meiosis. This is not a standard pictogram of meiosis. And that's why I say, they talk about this in the book. This is not drawn out. So if you've got questions, quite literally, now's the time, because this isn't in the book.
I'm just going to explain it to you.
Yeah, please do.
Okay, so the oogonium and the primary oocyte, they are frozen from fertilization till 15 years. Oh, okay, whatever. Once you're...
So nitpicky, this is frozen in prophase 1.
Okay.
Not that.
Okay.
And I know that sounds like me being a dick.
No, it's not.
But that could mean the difference between a right answer and a wrong answer.
Okay. So
It's just that this doesn't get repeated once we pause at birth.
Okay.
So essentially, this is up to birth, and this is once puberty kicks in.
Okay.
Does that make sense? So birth in the womb, puberty after birth.
So you're telling me that oogonium became the primary oocyte?
Yes.
And that's all the primary... You'll never go back to...
No. Everything will be frozen here at birth.
So mitosis only occurs before birth?
In the womb, yeah.
Okay.
In fact, not even this occurs once birth happens. Quite literally, once birth, essentially, this is going to be a weird way to do it. Right? In the womb, puberty.
Okay.
Does that make sense?
I'm going to do that as well.
But also realize this line that I wrote for womb is kind of funky. Because you still have primary oocytes frozen in prophase 1. So really the line goes like through it, but that didn't make sense.
Okay.
So I just wrote it above it.
Well, yeah, I just put the wrong one. Right here. Okay. That's all right.
Honestly, Whether you put it before or after primary oocyte, technically correct. And this is one of the reasons why I wanted to go through this one specifically, because it's so different and so, I don't mean funky in a bad way, I just mean funky and it doesn't follow our normal stuff.
Okay. And then when you say ootid and then ovum, what does that mean?
So when this divides, it's called an ootid.
Then it matures.
Then it matures to an ovum, but that's not a separate thing. It's still one cell. It's not doing anything different. It's just once you hit a certain maturation of it, it's called an ovum. The problem is that maturation is about that long. Because literally, right, fertilization happens. You get an ootid. Then it matures into an ovum before the nucleus of the sperm joins the nucleus of the oocyte. But that maturation process can be literally seconds long.
Okay.
So that's why we just put it as one quick thing, ootid then ovum, because it's not like there's a 3-week step in there.
So you rarely, okay, well you'll never have oogonium.
Once birth happens, yeah.
And you'll never have an ootid or ovum unless you fertilization occurs.
Perfect.
And those are your intermediates, and those only happen once you hit puberty.
Yep.
Okay, so it's pretty rare.
So, what's my N and my X here?
That is 2N, 2X?
Yep. So 2N, 2X. What are we here?
1N, 2X.
Perfect. And if fertilization occurs?
Are we still 1N, 1X or no? Yeah.
1N, 1X. So that brings up a completely different question that I have to ask you. So if I have an egg meeting a secondary oocyte, because the only way I have this division is for fertilization to happen, right? That means I have a 1N, 1X sperm meeting a 1N, 2X egg, which should give you a 2N, 3X resultant cell. We have a ploidy problem.
Yeah.
How do we not have a ploidy problem? And this can come up on the test. That's why I was asking.
Oh, I don't know.
Generally, the stumbling block is what people consider fertilization. Realize fertilization just means the plasma membranes of sperm and egg fuse. Has nothing to do with their nuclei.
Okay.
So what happens is their plasma membranes fuse. The egg kicks out a polar body.
That's where
Becomes an ootid, and as the sperm nucleus is moving to the egg nucleus, it matures into an ovum.
That's where the X.
And then you have a 1N, 1X nucleus meeting a 1N, 1X nucleus for a 2N, 2X kid. Does that make sense?
Yeah.
Fertilization just means plasma membranes fuse. I got an egg, I got a sperm, right? Their plasma membranes fuse. That's fertilization, and the second meiotic division occurs. It doesn't mean that the nucleus of the sperm has even entered the egg yet.
Okay.
So their genetic material hasn't met at fertilization.
Okay.
That's how we don't have a ploidy problem. Because when this division occurs and this thing kicks out a polar body, you have a 1N, 1X nucleus meeting a 1N, 1X nucleus, and everything's fine.
Okay.
Does that make sense?
It does.
That's what I mean by the definition of fertilization. Fertilization just means plasma membranes fuse, not the nucleus. That's how we don't have a ploidy problem.
Okay.
Does that make sense? Questions about any of that?
No.
All right, cool. So, the menstrual cycles, there are 3 of them, right?
Okay.
And I hate to say it, we need to go through them all. One of them is fairly straightforward, but there are some things we need to discuss at certain points and talk about. Some of these things are not addressed in the book, so we've just got to go through it.
Yeah, we're fine.
All right. Actually, completely drying off the board from cleaner tends to help before you write on it again. So the first menstrual cycle is the ovarian cycle. And the reason I want to talk about these, 1, it comes up a lot on the test. 2, guys don't really know or understand menstrual cycles because we've never had to deal with it. And 3, the menstrual cycle is actually 3 cycles, and each cycle is split into 3 phases. And so you've got to know all the details. So, phase 1 of the ovarian cycle is the follicular phase. The follicular phase is day 1.
Okay.
Because the menstrual cycle is a 28-day cycle. It's not a month long, right? It's not 28 days in February and 31 in December. It's 28 days.
Okay.
But, just like every other number in biology, that's an average.
Okay.
Menstrual cycles can be 25 days all the way up to 30, 31, 32 days. Right? Even standard body temperature, 98.6, not really anyone's body temperature. That's just an average. My sister's a total freak. Her standard normal body temperature is 96.8. When she gets sick, it drops.
Really?
She's weird, right? But every number in bio, it's just an average, right?
Mm-hmm.
So 28 days, but that's an average. Can be shorter, can be longer. Mm-hmm. What happens here is a primary oocyte is surrounded by a single layer of granulosa cells. The granulosa cells proliferate to form several layers and the oocyte secretes a protective mucopolysaccharide layer called the zona pellucida. And the zona pellucida comes back around, I don't know if we'll get there today, in polyspermy prevention. How do you prevent multiple sperm from fertilizing the same egg? So this will come back again in one of our later discussions. Okay. Then what happens, right, a mature follicle secretes, actually we'll even say it a different way. We'll say is surrounded by thecal cells and secretes lots of estrogen. And here's one of the times I want to step aside for a second and talk about this. A lot of guys think that the menstrual cycle is to prevent pregnancy, right? Mm-hmm. Woohoo, no baby this month. No, not at all, right? Realize the menstrual cycle is preparing a woman's body for pregnancy, not preventing it. So if we want to prepare the body for pregnancy, and we know that the fertilized egg and the placenta are going to grow and implant into the endometrium, first thing we have to do is build an endometrium to prepare for implantation. Mm-hmm. That's what this estrogen does, right? This is to build the endometrium. In preparation for pregnancy, right? Because the menstrual cycle is all about preparing for pregnancy, getting the body ready for it. Now, stage 2 or phase 2 is ovulation, or you could see it as the ovulatory phase. Either way. Now, here, ovulation or the ovulatory phase, this is around day 12 to 14, right? But everything's kind of on the ish category because nothing's perfect. What happens here, the follicular wall ruptures and a secondary oocyte is released into the fallopian tube. That takes us to the third phase. This is the luteal phase. And this is ovulation to day 28. Okay. What happens here is the granulosa cells form the corpus luteum, and it secretes lots of estrogen and progesterone. Now these two, we've already built the endometrium here. Mm-hmm. Well, we still have essentially 2 weeks before menstruation should kick in, so we still need to maintain what we've built. That's what these two are for. This is to maintain the endometrium, right? Because we're still hoping, the body's hoping for a fertilized egg, right? Because we're preparing. Now what happens is the corpus luteum degrades if new pregnancy occurs. And this is where we have to talk about something that most bio classes don't really talk about anymore. And that is, what is the corpus luteum? People, guys and girls, think it's just some magical anatomical structure that just appears, and that's not a thing. Mm. So we've got to talk about where is the corpus luteum, because you've got to know that for the exam. The corpus luteum is still in the ovary. Okay. So I'm going to draw a picture to help. Let's say that's a little oblong, but let's say that's an ovary. Right? We've got an oocyte with granulosa and thecal cells surrounding it. And when I have granulosa and thecal cells surrounded by an oocyte surrounded by those cells, that's what we call a follicle. Okay. All that together. Well, when ovulation occurs, the follicular wall ruptures. When that oocyte leaves, we can't call that a follicle anymore because the oocyte's not in it. So we change the name to corpus luteum. Okay. So the corpus luteum is what used to be the follicle when it had the oocyte. Now that it doesn't have the oocyte, we call it a corpus luteum. Okay. Does that make sense? Okay. So all we did was we changed the structure slightly when we lost the oocyte. Mm. So we can't call it the same thing, had to change its name.
So when it's it's a follicle when it's...
Yes, so when the oocyte's there, it's a follicle. When it's not, it's a corpus luteum. Okay. So whether they say follicle, whether they say corpus luteum, they're talking about the same place and the same thing. Mm-hmm. The difference is there an oocyte there or not? Okay. Does that make sense?
No, the oocyte will only leave no pregnancy occurs?
Well, ovulation occurs no matter what. Okay. Right? Because the follicle has to leave to get fertilized anyway. Mm-hmm. And so it's whether or not it gets fertilized determines if there's a menstrual cycle or if pregnancy occurs. Okay. Does that make sense?
It does.
Now, I'm going to refer back to this in a minute, but I got to erase it to put the next one up here. Yeah. Are you good? All right. So the next one, the next cycle we have to talk about is the uterine cycle. The easiest way to do the uterine cycle is with a graph. So I have endometrial thickness here. Right, how thick is the endometrium? We got days down here. Another real common misconception. Guys think actual menstruation, shedding of the endometrium, is the end of the menstrual cycle. It's not. It's actually the beginning, right?
Okay.
Because your graph of endometrial thickness looks like that. So shedding of the menstrual lining, the endometrium, is the beginning. We're going to talk about why, but we got to talk about the next cycle before we talk about that. But this is also divided into 3 phases.
Okay.
So your 3 phases are, and the days, you don't have to know exact days here.
Okay.
This is the menstrual phase. Right? This second portion, once it bottoms out, is the proliferative phase, named because the menstrual lining is proliferating very quickly. This is the secretory phase, so named for the sheer amount of hormones that are getting dumped into the blood at that phase. So let me know when you draw that, because there's some things we've got to look and point at and talk about. I'm going to see why I have 8 text messages from the same person when everyone I know knows that I'm teaching. So, hold on. The big thing here is that you've got to make sure you keep your phase names with the appropriate cycle.
Okay.
Here's why. Right? When we talked about the follicular phase, that was day 1 to ovulation, so that's about 2 weeks. That does not match up with the menstrual phase. Ovulation's only about a day. That certainly does not match up with the proliferative phase, right? This is the last 2 weeks. That's clearly not. So you cannot interchange phase names, which means if the question or the passage is talking about what's going on in the ovary, you certainly cannot pick any answer choice that says menstrual phase, proliferative phase, or secretory phase, because that's what's going on in the uterus. If they're talking about what's going on in the uterus, you can't pick anything that says luteal phase, ovulatory phase, or follicular phase, because that's the ovary. Does that make sense?
Yes.
Now, we can overlay the different cycles on top of each other, and I can draw a picture of what's going on in the ovary and all that, but the phase names, you cannot interchange them. The test is going to try to trap you into doing that. You've got to be careful, okay? Can I erase all of that? Because what we're about to do is, if the menstrual cycle comes up, it is more than likely going to be what we're about to do. Because the last menstrual cycle you've got to know is the hormonal cycle. And as much as this test loves hormones and the menstrual cycle, if you get to put the 2 together, they jump for joy. And so they like putting the 2 together. So I'm going to draw a big graph. Then we've got to go step by step. Don't draw this until I turn around and say draw this, because I got to make sure I get it right first. A little off. Let's actually complete that to day 28. Okay, that's close enough. You can draw that. Draw that.
Okay. Should I like save a lot of room or just enough for my...
You write small enough, we can probably... Oh, if you're going to draw the graph and write everything,
new page.
I go to a new page. Because I'm going to fill all of this space, and then even maybe turn around and go, okay, let me erase something else to add these last two things.
Okay.
I've even labeled the hormones.
Okay. Let's see how I do.
It's okay, it doesn't have to be
Do
perfect.
you want to do it?
No, I don't.